Okay, so we're in chapter 17, nutrients, and this is just a nutrient limitation experiment image from Milford Reservoir. And this is my second undergraduate student in 1990 or maybe 91, this was. And he's a professor now at La Crosse, Wisconsin. And this is a nitrogen and phosphorus addition here, which is together stimulate the most algae. This lake had modest cyanobacterial blooms, but now it has less than modest cyanobacterial blooms and is closed regularly. This chapter will talk about nutrients a little bit differently, how they're used and taken up, and then we're going to talk about stoichiometry for about half the chapter, so the relative rates of use of nutrients. And there's three ways you can think about nutrients getting into cells. And don't panic, these equations are fairly straightforward. And we can look at the first one, just straight uptake. And so the uptake is V, and it's a function of the maximum uptake and the substrate concentration. Could this be ammonium or phosphate? And the here. Substrate concentration, and these square brackets mean the concentration around a variable oftentimes. A half saturation constant, which is also a concentration, and then the substrate concentration. So what you can tell will happen with this equation is as the substrate concentration increases, this number gets closer and closer to 1, right? It'll never be exactly 1, but it'll get pretty close to 1. And as that happens, then, but when it's less, it's going to be less than 1. And so this basically means that as you increase your substrate concentration, V is going to approach Vmax, maximum uptake. The second one we can put in here is growth. So uptake and growth are two different things. And that's because cells can take up nutrients if they're limiting and pack them away. They have different ways to do that. With phosphorus, it's a thing called polyphosphate, and they can put a whole bunch of phosphorus, like 20 divisions worth of phosphorus into their cells, and then not need it for 20 divisions and then get more. So they can take up nutrients much more quickly than they can grow. So the growth is basically the same, that they can only grow at the nutrient concentration, but the growth rate is going to be slower than the uptake rate if nutrients are limiting. And then there's this equation called the Droop equation that links the concentration in cells actually. So you have to uptake the nutrient, get it into the cell here, and then from the cell you have to grow. So what really matters to the cell is the concentration inside of the cell, and the cell has to be over some sort of minimum concentration before the machinery works, and that's this Q0 at cell quota. And I will put these on graphs so you can see them, maybe. So here's the equation that we can talk about then this in more specific terms. This Ks is specifically the concentration at which the uptake is at half of the maximum growth, uptake is half of this maximum. So this is 1/2 of Vmax here, 0.02, Vmax is 0.04. You have to have 0 uptake at 0 concentration. And then we can look in the same thing with this equation for growth. This is the uptake, the growth rate at half the maximum growth rate. And then this one also has a similar, similar mathematics, the Droop equation, where you have this minimum cell quota. So in this case, 0 growth is actually above 0 concentration, and you have a maximum growth that it's trying to get to. And this will have to do with the way that organisms adapt to different nutrient concentrations. So there's a bunch of different things that can influence the uptake rates. In this case, we have light and we have nitrate uptake, and this is just expressed per unit amount of nitrogen per unit chlorophyll per time. And we see that while we have 0 light, we can get uptake of nitrate. The it increases drastically as the light goes up. And this is just from Flathead Lake. These are just bottles of lake water and using N15 labeled nitrate to look at the uptake into the phytoplankton. In contrast, ammonium is also sensitive to the light concentration, but not as sensitive as nitrate is. Can anyone remember think about why that might be? Right, so Tom got it. Ammonium assimilation takes less energy. Remember, they have to convert nitrate to ammonium before it can be taken into the cells, and that takes energy. And what's the other uptake, nitrogen uptake process from inorganic nitrogen that requires the most energy? Anybody remember that one? Ah, good, Lane. Nitrogen fixation. Good. So that's the order that they go in. So you can imagine that nitrogen fixation would be even more sensitive to the amount of light for a cyanobacterium if you are able to fix nitrogen because it requires so much energy. And then the bottom graph is a reflection of what Tom answered, ammonium assimilation taking less energy. So as the ammonium concentration goes up, the nitrate uptake rate goes down. There's the trade-off, right? The cells are, through evolution, have adapted when there's a lot of ammonium around to just shut down all the machinery used to take up nitrate and not feed it energy anymore, because the ammonium's cheaper to use and they can do that. Now, not all organisms do that. Some plants will, some plants use nitrate preferentially, and that's just because they never see ammonium really in appreciable concentrations in the environment that they've evolved in. So there's no reason for them to really have the ability to shut off ammonium uptake. They just concentrate all on nitrate uptake. And I think I jumped into my question of the day beforehand, so I'll have to ask you a different question of the day. So there it is. Look back at the nitrogen cycle for pathways of nitrogen uptake. So then we can think about what nutrient concentrations are in natural waters. And this is a bar graph, and there's total phosphorus under A and total nitrogen under B. And it's got the United States divided up into what are called nutrient ecoregions, areas that naturally have similar nutrient concentrations and vary from the other ones around them. So we can see Willamette and Central Valleys is up in Oregon and Washington some. Western forested mountains are throughout the West, where the forest, the dry West, Great Plains sits just to the west of us, north and south. Central cultivated Great Plains, that's pretty much where we are. Corn Belt, Northern Great Plains, that's Iowa. Further north is the glaciated dairy region, and you can just go through that whole thing and see that we pretty much go all the way to the southern Florida coastal plain and the Texas, Louisiana coast. The dark is the reference concentration, and the light is the current concentration. We have different tricks for figuring out what the references and currents are. But the first thing to take home is that some areas are naturally pretty low in phosphorus, and other areas are naturally a little bit higher in phosphorus, for example, the drier areas. And similarly with nitrogen, some areas are naturally low in nitrogen, some are higher in nitrogen. The second thing to take home is that the concentrations currently are way above those that were naturally there. So we have a lot more nutrient coming into these systems than historically. So now we're going to talk about how this plays out. So there's different nutrient concentrations out in different places. How do organisms adapt to those nutrients, and how do they adapt to taking them up? What has evolution given us here? So the first thing is a very general overview of the relative availability of nutrients relative to what are in cells. And so if you just take an average of plants and algae, and people grind them up and analyze them and see how much there is, how much nitrogen, phosphorus, hydrogen, all these elements in there. And since molybdenum is required, but in extraordinarily small amounts for most organisms, as a cofactor in enzymes, it's actually used in the enzyme nitrogenase to fix nitrogen and several other enzymes. So here's one molecule of that. And then they've taken all the bunch of different analyses from rivers and analyzed those. And again, normalized it to one molecule of molybdenum. So I can give you a question of the day. And if we look at cells and we look at rivers and why are oxygen and hydrogen the dominant molecules that we see in those environments? So here's a question of the day, so everybody can answer that. And then next, why is carbon not as dominant in fresh waters as it is in plants and algae? I'm going to try to pause here. Okay, good. Yeah, so you guys mostly figured this out. In water, hydrogen and oxygen are really abundant. You know, you just don't run out of oxygen and hydrogen in the form of H2O. In plants and algae, they also have water, but also all organic compounds have carbon, oxygen, and hydrogen, almost all of them, methane being maybe an exception. But since cells are made up of carbon, you have quite a bit more carbon in plants and algae relative to the amount of molybdenum than you do in natural waters. But because of bicarbonate equilibrium and CO2 and the fact that there are organisms suspended in world rivers and organic carbon and things like that, you still have quite a bit of carbon. Now we start getting into some of the other things. So there's nitrogen, a lot of nitrogen, a good bit of silicon in plants and algae. What algae are those that really need a lot of silicon? Good, Aaron, got it, diatoms. Anybody know of a terrestrial plant that needs a lot of silicon? Good, Cassie, grasses, exactly. Yeah, so actually there's a hypothesis that the grasses evolved silicon in their cells to stop herbivory and wear down the teeth of things that would eat it. And you know, like grasshoppers, if they eat grass, their mandibles wear out. And that freed a lot of silicon and it started entering the environment at much higher rates. And so the diatoms actually evolved after that based on that greater increase of silicon driven by terrestrial systems. So this is much higher amount relative than to what's inside of the, and phosphorus, there's not so much phosphorus relative to nitrogen and carbon in plants, but there's, in algae, but there's quite a bit. And it's so essential, you know, for DNA, RNA, ATP, lipids, all those things. And we can work our way down into some of the, you know, rarer compounds. So these up here are called macronutrients in general, and these are called micronutrients because they're needed in micro amounts relative to the other ones. I mean, you just don't think of boron being important in organisms, but we need it a little bit. And so then the last thing to do is that's really important as far as nutrient limitation is think about the average demand over the supply, right? So you just take the ratio of these two things to each other, right? So there's lots of water around in freshwater habitats, so that's not so likely to be limiting because supply demand is low. However, organisms do need carbon some, so the demand is a little bit higher than the supply rate is. But then when we get down to nitrogen, the demand is way higher than what's available. This is way higher than this. And the other one that's that way is phosphorus. This is way higher than this, right? So we get our first clue from this of what nutrients might be limiting primary production in freshwaters. And this is our first foray into stoichiometry. Stoichiometry is the relative ratios of nutrients, or anything, of any molecules, elements, I'm sorry. Okay, so one of the first concepts of nutrient limitation, and it looks like my slide needs a little bit of help there, doesn't it? is Liebig's law of the minimum. And I have law in parentheses because it's not, quite a scientific law in my view, but that's what it's called. And we're going to then go and talk about the case for multiple nutrient limitations. But basically what this came out of an agricultural setting. And what Liebig noticed was that the plants in a field are made up of all the same relative content. And they have, particularly if you have crops, it's all the same species. And they have a certain capacity to take up each of the nutrients. And given that capacity, they're not going to be matched exactly the concentration that's in any particular environment. So given that, and all things being well mixed and equal and temporally and spatially heterogeneous, that means over time and space they're the same. The will only be a single limiting nutrient, right? And if we look at the cell contents on general by moles of algae, for example, and if you grow them with all the nutrients that they can use, there's about 106 molecules of carbon for every 16 molecules of nitrogen and every one of phosphorus. So let's say that there's an availability of nutrients over here in moles per cell per time of 106 carbons, 4 nitrogens, and 1 phosphorus. So that's just what's available to them. Which of those would limit the growth of this cell here? Trevor, Tom, both got it. Nitrogen, right? Because this is in a lower ratio. There's more phosphorus than this, right? The phosphorus is available about 1/4 instead of a 16th, and the nitrogen also to carbon is such. Good. So we're going to play this game a lot. So one of the things that you hear in the literature traditionally is that in freshwaters, phosphorus is the dominant limiting nutrient. And in marine systems, nitrogen is the dominant limiting nutrient. However, when you go out and do bioassays, so we saw the very first picture in this lecture was of some carboys, and we had added some carbon to some, nitrogen to some, phosphorus to some, and nitrogen and phosphorus together to others, and looked how much chlorophyll yield there was. And we can do similar experiments in wetlands or in streams. You can use diffusing pot, something that diffuses nutrients out and the algae grow on. And we see what the nutrients that are limiting. We see that sometimes no nutrients are limiting. That's this bottom one, right? But sometimes nitrogen's limiting, not phosphorus. In lakes, oftentimes nitrogen's limiting in wetlands, and sometimes it's limiting in streams. And phosphorus is maybe limiting more often than nitrogen is in lakes, maybe limiting more often, or as often as nitrogen is in wetlands, and maybe equal, a little bit more than nitrogen alone in streams. And this is just a survey of a bunch of data from a bunch of different studies, so hundreds of studies. But if you look here, if you add nitrogen and phosphorus together, you really get a response in lakes and streams, not so much in wetlands as much, but you also have potassium or sodium potentially limiting. So any guesses real quickly why nitrogen might be more likely to be limiting in wetlands than in lakes and streams? And I'll give you a hint. It has to do with the nitrogen cycle, and there's a lot of anoxic habitat in lakes. Why are there separate labels for N and P and the N plus P tab as well, Lane asked. That's a good question. So in these experiments, you can add only nitrogen to the 3 carboys, only phosphorus to the carboys, or you can put both nitrogen and phosphorus in together, or you can have a bunch of mesocosms. So it's whether they're added singly or together. So the big response is when you add them together. Denitrification occurring, Aaron got it right. So wetlands are anoxic. There's a lot of denitrification there. So that's more likely to be, they're more anoxic than lakes and streams. So you might expect nitrogen to be blown off. Good. Well, what this says, though, is going back to Liebig's law of the minimum, that maybe not only one nutrient's limiting in one habitat. It could be different habitats, but why are we getting this co-response so often? And this brings us to Hutchinson's paradox of plankton. And as you saw, G. Evelyn Hutchinson he's the father of modern limnology. He is the person that moved ecology forward in a lot of ways, probably one of the most important ecologists ever, but written massive amounts of work on lakes and just comprehensive, an amazing man. So he wrote a paper called The Paradox of the Plankton. And what he said is we have these assumptions. All cells need the same nutrients. So we have that first graph a little while ago, table where they're in the ratios. Availability of those nutrients remains constant. So the supply is constant and everything is mixed. And he's thinking about lakes here. And then Hardin had this thing called the competitive exclusion principle. And that because of variability of different species of organisms, only one species is the best competitor for a limiting resource and will dominate. So if there's one species that's the best competitor for light, it's going to take over for all the rest if light's a limiting resource. And Liebig's law says only one nutrient's limits given those. Given that, if you go out to a lake, and even, particularly an oligotrophic lake where there's not much nutrient availability, and the last observation, most phytoplankton cells have about the same composition, which goes back to point one under the assumptions. So if you go to an oligotrophic lake, you'll commonly find hundreds of species of phytoplankton over time. And anytime you look at a lake, you'll see 10, 20. If you look long enough, you'll see more species than that. So all these things above here say that you should only have one competitive dominant. Yet the reality is that we have much more diversity than we would predict from those what seem like relatively reasonable assumptions. So let's have a little bit of a discussion on why, what things are happening in lakes that might, again. Okay, so all cells need the same nutrients? No. That we know that, for example, diatoms need silicon. Availability remains constant? No, we know that there can be a flood. There can be a fire that increased atmospheric precipitation. Nutrients evenly mix through the lake? No, nutrients flow in through different parts, like if it only flows into a river. There's seasonal stratification. There can be entrainment. There can be seiches. They can be mixing by wind. All those things can lead to unequal mixing. Only one organism is the best competitor for limiting resource. Yes, but no. We gave the example of ammonium nitrate and nitrogen gas. Nitrogen fixers, it takes them a lot of energy, but they'll have a source of energy of nitrogen that others can't use. Some may specialize on nitrate because they're more efficient with the energy, and some may specialize on ammonium. And then observation, most phytoplankton cells have similar composition. No, diatoms need a lot of silicon. And finally, there are species interactions that can knock down dominant species or give other species a boost. So mutualism might give, with a nitrogen fixing species, might give two species a boost over one that doesn't have that mutualism. Predation by viruses or selective predators that eat the dominant can knock down the dominant or give grazer resistant or virus resistant species the ability to come in. And so all these things play into each other, and we get lots more diversity than you'd expect with these simple assumptions. This is just a mental exercise, right? Because we know the reality is the observation that there's lots of species out there in your typical lake. But these baseline assumptions like Liebig's law maybe don't hold. And so that's the main point I'm trying to make. And this explains in part why there might be multiple limiting nutrients, which was where we started. Okay, and we've gone through a violation of these assumptions. And now we're going to move more into stoichiometry of primary producers and talk about remineralization, short-term processes controlling nutrient concentrations, So a dynamic view of nutrient limitation, of nutrient availability. And then talk about processes leading to remineralization and pulses of nutrients. So we're going to get more into this availability question. So we already talked about this typical concentration of algal cells being 106 to 16 to 1 if you grow them with plenty of all nutrients and some light. And this is called the Redfield ratio. It's named after a fellow named Redfield, who was the first who noticed it for the ocean. So if we go out and harvest cells from a lake or off the bottom of a stream and analyze them, we could see that the nitrogen is going to be less. We already answered this question, 106 to 5 to 1. If we go out and sample phosphorus, when phosphorus is limiting, now this ratio is greater than 16 to 1, and this ratio of carbon to phosphorus is also greater, right? It's twice as high as that. So in this case, phosphorus is half as available as nitrogen for carbon. And then we talked about co-limitation, right? When this is the case, cells can still photosynthesize and build up a lot of carbon, but the nitrogen and phosphorus stays at 16 to 1. And we can actually use this as an index of nutrient limitation. So rather than going out and doing these nutrient addition experiments, we can just scoop stuff up and throw it in the elemental analyzer or the other, whatever method we happen to be using, and get a really quick estimate of what resource is limiting nutrients. So where are these nutrients coming from? So we think about they're coming from outside the system, but if you look at the uptake rates of nutrients in a stream or in a lake, the nutrients that they are given the uptake rates would be just gone, and it could be gone sometimes for phosphorus within seconds. I mean, literally within seconds. And we know that the stuff's not coming in from the river, the atmosphere within seconds or minutes. We know that's going to be hours or weeks of time. And the reason that here's the place that we're going to come into this a couple times in this class, and that dissolved concentration of dissolved nutrients, ammonium, the phosphate, nitrate, is an amount. But the flux rate through that pool is a rate, and we can't confuse those. And we can see situations where phosphorus or ammonium, phosphate and ammonium, or concentrations are extraordinarily low in a eutrophic lake. So I started this lecture with a picture from Milford Reservoir of nutrient limitation in a lake that had pretty serious algal blooms in it already. When I used the radioisotope methods to estimate what the phosphate concentrations were in that lake, so it's the only way we can really get at true phosphate concentrations, they were extraordinarily low. They were at nanomolar concentrations. All the phosphorus was tied up in the biota. Any moment the phosphorus molecules available, it got snatched up. The phosphate concentrations were so low that when I did the assays, I tried to use what we called our ultra pure water, type 2 water from a purification system in the lab. That phosphate concentration was 100 times higher than was in the lake water. Evolution has driven these organisms to be very, very, very efficient at pulling up phosphate, much more efficient than some ion resins that people have synthesized to get most of the salts out of the system. Right. So a lot of years of phosphorus concentrations, billion years of phosphorus of competition for phosphate has led to extraordinarily efficient uptake rates of phosphate. And when you do these experiments and add the P32, the P32 that's in the lake water will disappear within minutes. It just disappears in the water. It's all gets taken up into the cells. So where is it coming from? Well, it's coming from regeneration or remineralization. This is a supply of nutrients from cells breaking, cells being eaten, organisms excreting, all those things, viruses break open cells, all those things can push nutrients back into the system. So on this graph, we have uptake rate, and uptake is here. Remember the Michaelis-Menten concentration, so this would probably be half saturation constant would be somewhere around here. And it's a function of the nutrient concentration. These processes like cells breaking or zooplankton excreting are happening at a rate independent of the nutrient concentration that's down here. So what happens is this turnover of this pool leads to some concentration of nutrient that's relatively constant over modest periods of time. The reason for that is that if regeneration is at a constant rate, if it's a negative uptake rate, it's supplying nutrients, right? And if you're supplying nutrients faster than organisms are taking it up, the nutrient concentration in the water is going to go up. If uptake exceeds this rate, right, then the nutrient concentration is going to be driven down. So we have this semi-stable state here. So what this says, two things, is that one, you can't know what this concentration is because it could be, these rates could be twice as high and these rates could be twice as high, and you still have the same stable point, right? And so that's the first thing that it says. The second thing it says is that the balance between regeneration and uptake is going to be extraordinarily important, that we can have that when you have high turnover. And just to get at some real data from a bunch of lakes and ocean studies, we look at uptake and regeneration measurements, right? They really are one-to-one. There's a one-to-one line. And in general, uptake and regeneration or remineralization of nutrients, whether it's phosphorus or nitrogen, ammonium, tend to balance each other out. I mean, they can be off by 100-fold, but in general, if you have a place where there's a lot of uptake, it has to be balanced by a lot of regeneration. Otherwise, all the nutrient would just be completely gone. There'd be no way to get it. Okay, so empirical data to show that I'm not lying to you. So things that lead to this remineralization, I've already talked about this a little bit. Some cells leak out materials, organisms throw enzymes out in the environment. Animals excrete all the time, either in pulses or in constant amounts. Heterotrophic bacteria, we'll talk about why they dump nitrogen and phosphorus out when they do. Viral infections cause lysis of cells. Nutrient mineralization tends to be dominated by microbial processes. So we'll get to the microbial loop in the next chapter, and we'll get at that. And in some cases, moving animals can assist nutrient import. And these include salmon, go to the ocean, grow big, come back, reproduce and die, dump a bunch of nutrients in. Hippos, go out and graze, come back in, defecate in the water. Migrating zooplankton, go down into the hypolemnion, maybe eat some stuff, go up, excrete, go down, up, like that. And also things that move across boundaries. So organisms that move through sediments can push nutrients up as well.